Hello all. Thank you for inviting me to make a presentation at the JST phenotyping meeting. I wish I could be in Japan where the temperature's a lot less than the 38 degrees Celsius we have outside at the moment. I'm going to talk about integrating phenotyping, machine learning, crop growth modelling and satellite imagery in plant breeding programs. And firstly, I'd like to acknowledge all of my co-authors. I won't read through all of them, but this is the culmination of a lot of research done with the University of Queensland, University of Tokyo, Purdue University and CSIRO, and more recently Wageningen University Research. I start off thinking about what breeders do. A lot of you have probably seen me talk about this slide before, but it's really important that physiologists and people working in phenotyping understand what breeders do. Breeding involves understanding the germplasm that you've got as the basis for the program, a cycle of choosing parents out of that program, making crosses between those parents, selecting the best ones and delivering that to industry, and importantly recycling these. And there's a lot of technology involved in the way that you accelerate germplasm through this process, and also in the way that you can do prediction. So one of the more recent technologies that we use is genomic selection, where you actually spend a lot of effort with high throughput phenotyping as one of the options in developing genomic selection techniques that allow you to predict parents from their useful parents from their DNA so that you can make good crosses, and also to predict good progeny that you might progress through the breeding program without doing any testing until you get to a later stage. You can find a large number of papers referring to phenotyping and its potential impact on genetic gain. Here's a 2018 paper from the team in Barcelona and CIMMYT who've summarised this fairly nicely, looking at the potential effects of genetic gain of phenotyping at different stages, when it can influence the phenotyping and the breeding program early, and it's affected by the size of it, so it's related to the cost of phenotyping. It can accelerate breeding cycles if we can select on secondary traits. It can make more accurate selection, something we're quite interested in Australia, so understanding spatial variability in the field. It can potentially accommodate genetic variation, which we needed to do to achieve genetic gain, and we can start to link these kinds of tools into crop and climate models, and we'll talk about some of that in this presentation. Machine learning has become a very big field. It encompasses a lot of different areas of work, and in fact a lot of things that used to be called statistical models somehow get associated with artificial intelligence. But it's separated into artificial intelligence and machine learning. Machine learning is usually thought of as things related to supervised learning, where you might have imagery that you label and train, and you're able to improve the models of how they detect objects. You've got deep learning, which is another method of doing that, which can accommodate lots of information accumulated over time, for example. Unsupervised learning, which is the type of things that we often used to call statistics, and probabilistic methods like Bayesian learning. All of these are techniques that you can read this particular review. These are various techniques that are being applied in phenotyping, and the types of things they're being applied to are on the right hand side here from that same review paper on phenotyping: stand count, spike count, yield components, disease monitoring, 3D reconstruction. Crop growth modelling. I think most people are fairly familiar with crop growth modelling by now. When I talk about crop growth modelling, I'm mostly talking about biophysical modelling, not structural or 3D modelling, which does get used in some areas of this type of research now. I'm talking about simulation modelling that's used across all of these different scales, from farm to region to national to global. And this is the type of model that requires inputs of rainfall, temperature and radiation. It models the dynamics of water and nitrogen through the season and keeps accountable of these growth variables. So it's the type of model that can accommodate information from phenotyping, and we'll talk about an example of that. Satellite imaging is another area that we're working on, and at the end of this talk I'll give a quick outline of a project that we're developing in this space, and these are all the locations where we're running this project. But here's an example of some of the types of imagery that you get. This is a trial that's about 200 meters long and about 50 meters wide. This is from Sentinel-2. This is a 10 meter resolution, 10 band satellite. And this is the same site. It's actually a couple of weeks later, and it's using the Pleiades satellite, which is a 0.5 meter resolution and 4 band. So we're starting to get to the point that with satellites we can actually see some of these plots in the field. So how do we bring these technologies together? One of the ways we've looked at this is in a paper we published a couple of years ago, working particularly with the team in Wageningen and some other partners, including University of Tokyo and INRA. And this paper that Fred van Eywick led talks about modelling strategies for improving the effectiveness of phenotyping techniques. So the types of things that we looked at were feature extraction, spatial modelling or statistical modelling of trials, the dynamic modelling of traits and the modelling of environment effects, and target trait prediction. So I'm going to talk about some examples of these 5 things, and hopefully you'll have some appreciation of those by the end of this seminar. This is a figure, a rather complicated figure from that paper. I'll leave it to you to read through it in detail, but what I want to do is take you through some of these examples of feature extraction, how we might use that in statistical modelling, and also use it to inform dynamic modelling. So we look at feature extraction to both estimate variability in the field and to inform our dynamic models. The types of platforms that we're using in our research include UAVs or drones, and also the Gecko tractor. This is a tractor that's got a modified spray rig on the front with a lot of different sensors on it. They carry these various sets of sensors. On the drones, we're usually using an RGB camera, sometimes a thermal camera, and a multispectral 5-band camera. On the tractor, we're using hyperspectral and LiDAR, that's push broom scanning. So we have very high resolution data of these plots that we drive over with the machine. So the scales that we get are multiple pixels per organ. We can scale that up to canopy, plot and field, and we built a pipeline to do that. And here's one of the pipelines, the aerial imaging platform that we developed at CSIRO. That platform's got a technology to allow us to estimate crop cover, 3D lodging, canopy temperature, and we reviewed all of that in a paper a few years ago. We actually started research in that space in about 2008, and we just have a summary of a pipeline that's become pretty commonly used by most people by now, that allows us to process imagery and get results at the plot level. We have another pipeline at UQ, so the Phenocopter pipeline developed at CSIRO, and at UQ we have a different pipeline. It looks much the same, but it's designed to process the information from the drones and from the tractor system, and it has a system called Gecko Explorer that allows us to explore all of the data sets and it also allows us to start doing analyses. So we have tools built on top of this, for example, to estimate biomass during the season or from any particular date. So if we have observations of biomass and we've got data from the tractor, then we can process different wavelengths or hyperspectral information to try to predict biomass, and then process that through the system to estimate things like radiation use efficiency. So how do we use these platforms to estimate variability? Here's an example of a wheat breeding trial. It's low yielding. It has 2 to 4 hectares, 2 to 4 tons per hectare. And you can see this particular trial's got some problems running through it, rhizoctonia affecting the crops. So this is an intrinsic disease or something in the soil that's spread through here. And so then within each of these plots, so these plots will get harvested by a single harvester at the end of the season. But within these plots, we're interested in these bordered plants. These are the ones that we're trying to estimate the yield in. And we've got other plants, these unbordered plants and compromised plants that are near the edge of whatever this disease is. And then there are also gaps in these red areas where there are no plants. So one of the questions we have is whether we can do pixel-based analyses to account for these and recompute the harvester result, or whether we can take other statistics like a ground cover estimate and use that to adjust the final yields. So here's some examples of where we've got gaps in the field trials when we're screening. So this one is the rhizoctonia disease I just showed you. It's actually from a trial where there was a phosphorus treatment as well as a genotype treatment. It's a rather large trial, 1,600 plots. And there's some other data here from a breeding trial from a Longreach company with 5,000 plots. So we're able to process these kinds of data through Phenocopt. And if we start slicing a plot up in 2D, these are particularly bad plots. I think most people would pretty much eliminate these plots from any kind of analysis, but it's to give you a feeling for the variability here. If you look at the ground cover here and you estimate ground cover and slice this plot up, you can see that there's variation in ground cover across the plot. And that's at York, and this other one's at Piney. So these are the two sets of trials I just showed you. And so by slicing these up, we can actually investigate variability within the plot as well as across the entire field and see whether we can adjust for that. Here's an example where we've estimated the gap frequencies in the plot. So if we've taken what proportion of these plot sizes are less than 0.2 in coverage, so less than 0.2 of the pixels are covered by plants. These photos were taken in the late vegetative stage, so we really would have expected the crop cover to be up around 70–80%. And you can see that in this is the proportion of plot sizes that are less than 0.2, and this is the count of the number of gaps that are in these plots. So we've got 150 of these types of gaps that are less than 0.1 or 0.1, and that was out of 1,600 plots. So about 10% of the plots were actually had less than 0.1, and if you look at less than 0.2, there was almost 15% of the plots that had this poor coverage. So one of the ways that we can deal with this is to use some software. We're using this software from Wageningen. The Wageningen team have analysed this, and the software is an R package called SPATS. that package allows us to fit regression models to the plot data. So this is not analysing the within plot data. We are going to be looking at that, but at the moment this is the plot level data. And these are the fitted yields. And we're getting a heritability of about 0.8 from this analysis, which is good. So you can see in this trial there's a fitted spatial trend. So there's lower yields in this part of the field and they're increasing as we go across the field. So this is from the yield data, that's the raw data, here's the fitted data and the residuals, and these are the genotypic blubs. So you can see we've got a pretty good normal distribution of the blubs, which is the kind of thing that you'd expect with a trial. It's got about 800 genotypes in it. So this is, and with a heritability of 0.77 computed from this data set, that means it's a fairly reliable trial. We also looked at fitting the ground cover. So that Z mean just means that there's been some standardization of the ground cover. That's the percentage of pixels covered by vegetative component. We've standardized that somewhat across these trials. I won't go into the details, but you can see we've picked up these spatial effects from ground cover, which broadly map against the yield. Again, a fairly good normal distribution, high heritability. So it seems that ground cover during the vegetative period was also spatially affected, but we can model it quite well. And then when we compare these, so here's the yield data you just saw, here's the ground cover you just saw, we can see that there's a correlation between this ground cover and yield, which is about 0.52. So we know that the ground cover variation accounted for about 50% of that yield variation. And this allows us to try to control for this kind of trait and recover a trial that's probably been grown in a less than optimal place. So how do we use feature extraction to inform our models? Here's a crop model. This is the approximate processes that are in the AppSim crop model. We have all of these environment inputs that are daily inputs: radiation, temperature, photoperiod, VPD, rainfall. We have a series of parameters, things like leaf appearance rate, extinction coefficient, radiation use efficiency, and some traits associated with roots and water uptake. These parameters affect the phenology, the leaf production, leaf number, flowering time, which influences the expansion, so leaf area index. That influences the amount of light that's captured by the crop, the nitrogen plus the dynamics of the canopy light interception affect the radiation use efficiency, and these come together to influence the crop growth rate that's dependent on radiation. And then on the other side of it, we look at the water supply, the amount of water the roots can access, the effective transpiration efficiency of this crop, and what its potential crop growth rate is given the amount of water it has. And these things influence the actual crop growth rate and the biomass around anthesis, and then that growth rate around anthesis influences grain number and sets up the grain number. And then there's during the grain filling period, and you can see this is the different sort of stages of what the crop's doing. During the grain filling period, grains grow with a relatively constant rate, and depending on the grain number and the grain size and the residual carbohydrate and the biomass through photosynthesis directly, we have an influence on grain yield. So we're able to capture all of these processes, but if we think about situations where it's irrigated, we're mostly interested in the phenology, the canopy development, and the light interception. In the past, I've talked a little bit about machine learning and applications of crop growth modeling. There's multiple applications for this type of technology in filling in weather data, estimating soil characteristics and characterizing stress indices. These are the data sources for those types of applications and some examples. I won't talk about those. We're going to focus more on these parameters for crop models, so phenology, leaf area and radiation use efficiency, how we can derive these from imagery and estimate flowering time and genotype parameters. And then we'll also talk about parameters for grain organs and yield, because we're starting to use that information in a crop like sorghum to do head counts and grain sizing. And head counts are important in the sorghum model because head counts influence the amount of tillering that we get. So going from that model that I showed you just before, here's some work that we've been doing in Purdue University, and we're looking at measuring leaf area index and estimating fraction of light interception. That's the K extinction coefficient. So we're interested in how that extinction coefficient varies across genotypes. We're also interested in estimating the radiation use efficiency, but to be able to estimate this, we first have to be able to estimate the leaf area, and we use measurements. In some cases, they've got to be physical measurements of the largest leaf or the size of the largest leaf, so that we can rebuild this pattern of potential leaf size. And if we know the leaf appearance rate and the final leaf numbers, we can calculate the phenology. So that allows us to produce a dynamic through the season of the total leaf area. And if we know what that leaf area is, we can calculate the light extinction coefficient. We've got the light extinction coefficient, we can estimate the light interception. And if we have that information, we can calculate a radiation use efficiency. So that's just the amount of radiation intercepted versus shoot dry weight done from experiments. And one of the things that we're working on, particularly in the Queensland work, is how do we estimate the shoot dry weight indirectly using a drone or something like that. So here's some recent results that are just in press that Kawei Yang at Purdue has put together, and in that paper that's about to come out in silico biology, we were able to fit these leaf area curves for 18 diverse genotypes of biomass sorghum. You can see we can model the patterns of leaf area index over time reasonably well. We can capture the extinction coefficient. So this canopy cover has been estimated from a drone or imagery above, and we've compared that to leaf area index, and you can see that we can estimate this extinction coefficient and a radiation use efficiency. So I haven't shown the RUE here, but here we have for 18 different genotypes grown at about a dozen sites and in multiple years. This is validations against independent data from Texas and another site, and we're able to estimate the biomass change over time. So the next component is counting heads, trying to count heads in sorghum to estimate tiller number and leaf area, because the head number is actually part of that, or the total tiller number is part of the estimation of leaf area. This is work that we're doing in another GRDC project, and it's been undertaken by Yanyang Gu and Crispin James at UQ, and of course in conjunction with Wei Go, who worked with us over the last few years to develop this labeled sorghum data set that I referred to earlier. And this is some recent work where we've been looking at two different types of machine learning techniques to detect, or three types, so detection-based ones, efficient det, mask RCNN, and a density map estimation, CSRNet, which is a different methodology which tries to estimate the probability that there's a head at any particular pixel in the image. So we've tested those techniques with that data set, and a comparison of the histograms shows us that the CSRNet, so this density-based approach, is actually rather good. It's well distributed around 0. It has a small error less than 5 heads in these images that had around about 100 to 120 in each image. And what we find with the object-based methods, efficient det and mask RCNN, is they always underestimated the heads. They always missed some of them. I mean, the error wasn't terrible, but one of the questions we have is what's the error look like when we look at independent data? So when we looked at some external images, these were rather challenging ones. We compared their performance against these external images. So you can see that CSRNet was the density-based system was actually the best on the test set. But if you accept that efficient det got most of the images, most of the heads in these 3 images, well, actually mask RCNN did a better job because there were around about 100 in all of these. So this 3rd image was quite difficult to measure. But you can see that CSRNet really didn't do so well. It works very well on test and validation sets, but it doesn't generalize as well. So CSRNet is the type of density approach that's behind things like TASSLNet, but it struggled with these independent data sets. And I think these are the same kind of results that our French colleagues at INRA and ARVALIS have found for wheat. So Crispin and Yanyang have done a little bit more work on this. If we started to think about the distributions of head size and whether we could start to manipulate these labeled images. So we have about 120,000 annotated sorghum heads that were annotated by the students and team in the University of Tokyo over the last few years. And here are some examples of larger heads and smaller heads. And one of the things that we're interested in is whether we can extend the training by using GANs. We've got a big variation in head color. So one of the things that Crispin has worked on is used these generalized adversarial networks to generate fake images. So we could change these brown heads into white heads and then go back and see if we could test the model and generalize it better so that it could handle different colors of heads. And this has worked rather well, which has been really useful because we didn't have so many images that were white heads. But of course, if we've already labeled them as brown and then they're white, it's a lot easier to use those images than it is to have to label new ones. So these applications of ML and sort of crop growth modeling and phenotyping include things like being able to extract objects and things like trying to estimate crop cover and leaf area index using decision trees or semantic segmentation. And we have a research project in that area. But the objective of doing those types of things is really to get down to estimating other parameters that might be more genetically controlled. So things like radiation use efficiency, transpiration efficiency. So I've sort of covered those 3 aspects of feature extraction, spatial and dynamic modeling, and I'm just going to mention in a couple of slides some previous work in these other 2 areas before I conclude with a description of a new project we're working on. So this is some very old research that we did on sorghum in Australia, where we were running computer simulations of sorghum growth with a historical weather record. You can see that sorghum production in a location in Queensland, average yields of around 2 tonnes per hectare. That's what it is in this part of the world. And you can see we have a very large annual variability. And if you take each of those seasons, you can construct a stress index, which comes from the APSIM model. So if we run the model at each one of these points, and these are model yields, in fact, you can see each one of these years comes up with a stress pattern. And what we did in those papers, it was some of the original work in this area, was to summarize these, so we apply them through a sort of a cluster analysis and create what we called environment types. So here are the 3 environment types that we derived from an analysis of all these locations that span about 1,600 kilometres from the top part of the sorghum production zone to the bottom. And we found that there were these 3 types of stresses that occurred in about equal proportions through the seasons, across any combination of soil type and season and location. So there were these types of stresses that we called a mild terminal stress that occurred about 40% of the time. And there were these severe terminal stresses that occurred about 35% of the time, and mid-season stresses where the stress got relieved that were about 30% of the time. So while these occur in about equal proportions, they don't necessarily happen each year. So you might not get that exact proportion in any particular year. And I'd refer you to those papers to have a look at some of the discussion we did around that. So some of the research that we did from that was to then look at artificial genotypes. So these were simulated genotypes. And in these 3 different types of stresses, we would see different levels of yield for the whole panel of genotypes. So for example, you could see that early flowering genotypes did better in the severe terminal stress, but only moderately well in the mild terminal stress, and in a mid-season stress they were actually quite low yielding. So this is a sort of response surface if you were able to plot the entire possibilities of a breeding program. And one of the things that we've done is we've run that through a breeding program or a virtual breeding program and looked at how genetic alleles get fixed over time if you're selecting in these types of stresses. So this is if you had different types of repeatable stresses, and this is if you sampled randomly across the environments. And what the important thing is to see here is that if you did have a series of severe terminal stress environments, you would select for early phenology, and you would select for high TE and an osmotic adjustment, which was associated with stem retranslocation, and you would then select for stay green. So these traits would actually be selected at different priorities depending on what stresses you imposed in the breeding program. And this is something we could study with the model. So the last thing I wanted to mention was this new project that we're working on. It's a collaboration between UQ, Wageningen University Research, and CSIRO. It's called INVITA and stands for Innovations in Variety Testing in Australia. We've shamelessly stolen this name from our colleagues in Europe who have a program called INVITE, where this is Europe instead of Australia. And GRDC saw the opportunity to invest in a project that complemented INVITE. And so this is actually a sign of respect of the INVITE project, in fact. And we have 3 different work packages here. One on data augmentation, where we're collecting data with cameras in the field. They're watching the plants grow, basically. Canopy temperature sensors, which are providing a canopy temperature index. Some sensors that measure multiple spectra. And we're also monitoring with satellite in all of these locations. So we have these types of measurements in about almost 100 trials across Australia this season. And that's part of the data augmentation. In the analytics, we're looking at how to account for plot-to-plot variability, as I talked about earlier, and how to account for G by E. We're working on that as well. And we have a third component where we're using simulation and machine learning to try to test out new ideas and new ways of improving our prediction of variety performance. And that sort of complements this work our partners are working in HTTP tools in INRA and Arvillus and their work package 2 in the Invite project, and in work package 4 in the Invite project, Bargaining is working on predictive modelling, and they're involved in our work package 2. So these packages are all working together. These are the Australian packages on the left, and we're starting to make some progress and some appointments in that work, but hopefully I'll be able to update you on it in a future JST meeting. So hopefully I've shown you some aspects of how we think we can bring these technologies together. We've got research in wheat and in sorghum in this space. The wheat work is national, so we're able to collect a lot of data in that space and start to test some of these ideas. And we're hoping that we'll have some nice methods to do that in our drought limited environments. So thanks again everyone for allowing me to give you a bit of an update on the type of thing that we're working on, and look forward to seeing you soon. Thanks a lot.